Hello, I'm Litvia Kolaku. In this project, we are doing homemade agar preparation and surface sampling. For this project, we need 1 cup of water, 1 teaspoon of sugar, 1 teaspoon of gelatin, 1 teaspoon of beef extract, and 2 petri dishes. Firstly, we take the water, we boil the water, and then after 2 minutes, after the water is already boiled, we add the sugar, the gelatin, the beef extract. Then we leave only 1 minute until everything is melted, and then we fill the agar plates. In this project, we will fill 2 agar plates, half of them. Then we leave in the fridge for 5 to 6 hours. As we see already, I left them for 5 hours on the fridge, and now the agar plates are ready with it's already done. Now I have to do the surface sampling. For this one, I need the cotton swabs, in which I will just get the microbes, let's say, from different parts. As my partner will do the sink, I will do the computer keyboard. The surface sampling is already done. So in one plate is signed that is from the computer keyboard and the date, and then the negative control. We need the negative control to compare those 2 plates. And now we leave in the room temperature 3 to 5 days, and then we will see the results. Here we are on day 7 with the 2 agar plates. The negative control we see has no growth, is the same as it was on the day 1 before incubation. These are only the beef extract pieces, and here we see that we have some growth. We see that the plate is contaminated, so the bacteria has grown here, as we see better. Bacteria, as we see, the color of the media has no change. The bacteria, the colonies, have a color like brownish color. It's a little bit different from the results that were expected from the protocol that I followed. It should have been more growth. Anyway, maybe because of the incubator that we don't have at home, maybe that's the reason. The colonies, that doesn't look exactly, but it seems like the shape is irregular. Margin arrows, elevation convex, and order is pudrid.
My name is Mukul and I am the student of biotechnology program, and topic for this project is preparation of media from home ingredients and its optimization. And to check its viability, we'll be doing surface sampling. So what kind of materials I'm using are bottled water, measuring cup and spoon, gelatin, salt, sugar, a saucepan, some beef broth powder, sampling swabs, and some petri dishes. So what I'm gonna do is I'm gonna take 125 ml of this bottled water and half tablespoon of gelatin, half tablespoon of beef extract. I'm gonna measure it with this, and half tablespoon of the sugar and some salt, so that osmotic balance could be made between media and the cells which I'm going to grow. And this saucepan I will use to heat all these ingredients and mix them all. So ingredients which I've mentioned earlier are my variables. So all those quantities which I'm going to use are my fixed variables. And the method is I'm just going to take all the ingredients, mix them in the pan and heat them up, and then I'm going to plate it in the petri dishes. So I cannot film while I'm mixing and preparing whole thing on the gas. So what next thing you guys will see when I will plate my media. The media is perfectly set. So in previous clip I have shown you that my media was perfectly set, and then I sampled it and then I incubated it. So I incubated it in this box over the week actually, and then as you guys can see, this plate was my I kept as my negative control, but unfortunately my negative control failed, and this plate on which I have sampling, as you guys can see, there are various things, and I don't know in cam, yeah, it's visible. As you guys can see, there is certain kinds of turbidity, and you can clearly see the difference between my media set and now my media, which is after 1 week. As you guys can see, there is some fungi like growth, and there are also some colonies here. Yeah, now you can see, right? And that was my control, but unfortunately it failed, and cause it has growth on it and it's not the same. So yeah, so far it was we are able to make some viable culture at home with home ingredients.